Hi, and welcome to the presentation of my infographic titled Fine-Tuning Non-Coding RNA to Halt Virus Infection. So we've been taught very early on that in biology, the information flows from DNA to RNA and then proteins get made. But actually, only a very small percentage of RNA makes proteins. So the question is, what happens with the rest? The rest is called the non-coding RNA because it doesn't encode for proteins. And the non-coding RNA is a part of the dark matter of the genome, which is basically a very cool name for unclassified or poorly understood genetic material that has gained attention only in the recent couple of decades. So even though non-coding RNA doesn't encode proteins, it still regulates their activity. And it does that through interactions with other molecules in our body, and these interactions turn certain genes on or off. One such molecule is the subject of our interest, and it's called the circular RNA. The circular RNA has its head and tail joined together to form a continuous loop. And circular RNA is also capable of interacting with different other RNAs or even proteins. And we're interested to study these interactions in the context of the virus infection. So how do we study circular RNA? We can study it by using supercomputers and specific algorithms that have been developed to pick up on a signature which is unique to the circular RNAs. And thanks to the supercomputers, now we know that circular RNAs are not only present in people, but they're also present throughout the tree of life. So domestic and wild animals also have them, plants, and even very small and primitive organisms such as viruses. Another way to study these molecules is in a lab, which we also do. And before we can study them, because they're so small, we need to visualize them. So we can mark them with fluorescent probes, which then we can visualize using different machines, such as a flow cytometer. Another thing we can do is study the effects of these molecules on genes and consequently on the infection itself. And we can do that by turning certain genes off or even turning certain circular RNAs off. For our model, we are using a human immunodeficiency virus, or HIV, which is the causative agent of AIDS. And so far, by using supercomputers, we've discovered that certain genes promote virus spread, and these genes actually make more circular RNAs in cells that are infected with the virus. We don't really know what that means at this point, but it's certainly a very interesting find. Another thing that we did is we've created an artificial circular RNA molecule, and we've created it in a test tube. And we can put these molecules into infected cells and monitor what happens. And what we noticed is if we put this artificial molecule, the infected cells actually get help to fight off the virus. We think that is because this artificial molecules act as a decoy for proteins that the virus itself would usually utilize to promote its own spread. If you want to learn more about the hidden universe of circular RNAs, you can scan this QR code in the lower right corner and dive with us into the rabbit hole. Thank you very much for your attention.